Okay. I think I just need to. Okay, so we were looking at the steps of gastrulation in early development in frogs. So what we have in here is the different stages through once we have created our, where's my pointer? That's not the one I want, this one. So here we have our animal pole, we have our vegetable pole, okay, and then in gastrulation, remember, if it's not gastrulation, it's not an animal, right? And so in here, what we have is the different movements that allow the ectoderm, always in blue, and the endoderm to take the position to allow the endoderm to place in between the two layers. So we have the animal pole, the vegetal pole, and we have the blastocele. Same thing as we saw in sea urchin. And excuse me. This blastocele plays an important role in separating the dorsal side and the ventral side, at least here at the beginning. Okay, so the anterior, posterior, dorsal, ventral, right and left axis normally are specified by the events of fertilization, as we saw before. Remember the formation of the great crescent that will allow the anterior, posterior portion. And all of these axes are created once we get into gastrulation, not before, but just during gastrulation. So let's try the general movement pattern of these germ layers. So here we have at the edge, in between the animal pole and the vegetal pole, we have a section, in the posterior section here, that we're gonna see some things called the superficial cells. These cells gonna start the first movement, similar manner as we saw in sea urchin, is the invagination. Okay, so these cells gonna start invaginating, gonna start poking in at that layer here between the animal and the vegetable pole. Okay, and this is what are gonna start generating the archenteron or the gastrocele. Okay. So we have invagination, and once the cells start invaginating, as we see here, we see the appearance of what we call the lip. And this will be the blastopore. Okay, so this is the dorsal lip of the blastopore because it's going towards the dorsal side of the animal. Okay, so remember, we're going to have cells moving either way, right? As we see here, it goes both ways and continue to invaginate. This is the blastocele that now start getting displaced towards the left. And here we see the appearance of the archenteron. Now, the cells that are placed here at the edge and start invaginating, the ones that are leading. So these cells here, you see how they change shape a little bit, the ones that are leading the archenteron? These are called the bottle cells. They change their shape and become now the bottle cells. These bottle cells line the archenteron as it forms. So start going in, and they're going to be lining this archenteron as they pass through the lip, the dorsal lip of the blastopore. So it's very similar to what we saw in the sea urchin, right? Some cells that were invaginated and were reaching to the roof of the archenteron, right? So in this case, the invagination of the cells initiates the archenteron in the same manner in the frog. So if we look at now a cross staining slide, we see here the blastocele, okay, and that gets displaced, and now this is the archenteron or gastrocele. At the end, we see here what we call the plug, okay? So now, one difference with the seed version is that gastrulation begins not at the vegetal pole, right? As we saw, there was in seed version was down here. In frogs, it starts as the marginal zone, okay? And so more at the equator between the animal and the vegetable. Just keep that in mind. So here, the endodermal cells are not as large, and the yolk at the bottom. So this is, you see this, the cells here, these marks are tiny, while at the more vegetable pole, they are larger. So that's what allows these small cells to start invaginating. This big cell wouldn't be able to do so. So this marginal zone is really important for allowing the invagination of the cells and the formation of the archenteron, okay? So as these cells start getting in, the marginal cells start involuting, okay? Remember what is the movement of involution? Is the rolling over of a layer of cells over that lip, and this is what we see here. These cells start rolling over, you see? Okay, so start involuting, okay? While the ectoderm, the cells that we have at the edge, are doing what we remember how it's called this movement that goes all the way around the egg? How is that called, that movement? Annalie? You remember?
I'm not sure if it's something like envelope or I don't know. I don't know what it is.
It starts with an E. Epiboly. Okay? Epiboly is the movement that goes all around, is covering the whole thing. Okay? So cells at the margin invaginate, cells that are leading and are coming here are involuting, okay? And then the ectoderm is doing epiboly, okay? So the movement of the sheets are around, that spreads all the way around until we get into the end, okay? Where we're gonna converge together and form the blastopore. So this is the blastopore at the very end. Are these movements clear now? Yeah, okay, perfect, thank you. So the anterior, posterior, and dorsal and all that follow those same movements. So we have the epiboly, they start going and stretching. We have the vegetal rotation with the blastocele roofs are going from the animal towards the vegetal section. We have here the invagination of the cells are going inside for the formation of the archenteron. Here we have the transformation of the cells that are leading the formation of the archenteron, that are the bottle cells, okay? And in here we have the involution. Here we see how we start looking at the migration of these cells, okay? And at the very end, we have the convergent extension movement. Okay. So the epidermis, or the outer layer of the skin, is stretched over the entire body and originates exclusively from the animal hemisphere. This is the same thing that we have said before. So start going for epiboly, and it gets into the closure that it gets right here. So as seen here in figure A, during gastrulation, the ectoderm is pushed, it gets thinner. So here we see how it starts changing, it starts getting thinner and thinner as it surrounds completely the embryo. Now, in order to stretch, two things must occur, okay? One is an increase in a number of cells through cell division. Okay, so we're going to have a layer that's going to allow the division of cells. And second is the integration of several deep cells layer into the upper cell layer. So here we see how these ones that were at the bottom now become just underneath as they start stretching and moving. Okay, so time by time we see 3 layers, and now we have a mix of these layers. Okay, so SL stands for superficial layer, and DL stands for deep layer. Okay, so cells from the deep layer sense protrusive processes radially towards the surface. We don't see them too much, but maybe in this one here, we see how it start pulling themselves up with little like anchors. Okay, and that's how they're going to start intercalating. So in here, if we look at it, we have dark and lighter. So they start intercalating at this level. Okay. So we have all those things happening. So thinning of the layer due to the mitosis, cell division that needs to increase because those cells cannot just stretch to the infinity. Okay. So they get replaced a little bit. Okay, so when the migrating marginal cells reach the dorsal lip of the blastopore, so now, oops, let me just unload. So we came all the way down and we reach back again, that dorsal lip. They're going to turn inward and travel all along the inner cell. And at the end, the leading process of that ectoderm becomes the ventral blastopore lip. So we have now the dorsal blastopore lip, which is the point at which the cells start invaginating to form the archenteron, and the opposite end now becomes the ventral blastopore lip. Okay. The cells making up the lip of the blastopore are constantly changing as they start gaining the inside. Okay, is this clear? Okay, so and here once again, migration by Epiboly, invagination, and involution of cells so we can form the archenteron. And once we have these 2 layers, ectoderm and mesoderm, ectoderm and endoderm, excuse me, the mesoderm indicated by red start placing themselves in between the 2 layers, endoderm and ectoderm. Okay. Now, at the very end, the section between the ventral blastopore lip and the dorsal blastopore lip is what we want to find the yolk plug. Okay. As time passes by, we want to see the differentiation of that mesoderm into here we have the mesenchyme to form other parts of that embryo, as we're going to see later. Okay. So the first cells to compose the dorsal blastopore lip are the bottle cells, right? As cells start invaginating. Okay, and those are the ones that form the leading edge of the archenteron. These cells then will become the pharyngeal cells of the far gut, just because as they start going in, they're going to be placed, as we're going to be seeing here, a little more towards the back of the embryo, right? So those leading cells, bottle cells, going to become the foregut of that embryo. As the first cells pass into the interior of the embryo, as we're going to see right here and right here, the dorsal blastopore lip become composed of cells that involute, and that's what we have the rolling over of the cells. Okay, so they start involuting, and this is going to form what we call the precaudal plate. Those are precursors of the head mesoderm. So that's what they become a little red color here. So I'm gonna go back, and we see that as they start going in, they become a little more of mesodermal cells. And that is by involution, okay? Then the third type of cells involuting into the embryo through the dorsal lip are the chordomesodermal cells. And keep an eye on these guys, the chordomesodermal cells, because they are very important later in development, okay? These are the cells that will form the notochord. And as I said before, the notochord is a very important structure. It's a transitional tissue, but very important in development. These are tracial mesodermal backbone, important in inducing and patterning the nervous system. So if something happened to these chordomesodermal cells that form the notochord, well, the whole thing will be kaput, okay? So as the new cells enter the embryo, the blastocele is displaced towards the side, and in here we're going to have the formation of the gastrocele until we have at the very end the blastopore. So this is the beginning of that lip. It seems kind of cute, like somebody's putting the lip towards the mouth, okay? So while the blastopore lip extends laterally and ventrally, so here we have the beginning of this dorsal lip, we have the Epiboly that is going inside, moving all around. And here we have a little bit more of the dorsal lip, and as it progresses, we have the bilateral lips. And at the end, we have the ventral lips. So dorsal lip, lateral lips, and the ventral lips. And all of this then happens because of the evolution of cells. So we're going to have a widening at the beginning here. A lot of the widening of the blastopore is more like a crescent here to form those lateral lips. And finally, the ventral lip, which additional mesoderm and endodermal precursor cells are going to pass through, okay? So with the formation of the ventral lip, the blastopore, has formed a ring. That is what we have in here. So we need all four lips to start forming, the two lateral, the dorsal, and the ventral. And then now the yolk, which is the endoderm, is completely inside of the ball, right? So, and because now all the yolk is inside, that's what we call this the yolk plug. Okay, we're gonna come a little bit more internalized with time. Okay, and at this point, the endodermal precursors had been brought into the interior because before were only in the vegetal plate or hemisphere. Okay, the ectoderm is completely surrounding that embryo, and the mesoderm has been put inside. So now we have a tryphoblastic embryo that has the 3 germ layers into the right place. Okay. We see here, we see all these little cell divisions are still happening, right? So that's how it's still stretching. Now, one of the most fascinating aspects of gastrulation in frog and fish, as we're going to see later, is the Epiboly of the animal cap and non-involuting marginal zone cells. Okay, so this is a very important term. The non-involuting marginal cells is the NIMZ, and you want to see it appear later. Okay, because this cover completely the egg. So at the end, the ectoderm will completely cover the egg surface, and this is achieved through a rapid cell division, as we saw prior. Now, a second step that is important during Epiboly requires the presence of a molecule that we have already seen, the fibronectin. Okay, so fibronectin is what we can see in this figure over here, and that is guiding the cell movements. Okay, So it seems that fibronectin is used as a highway to guide the ectodermal cells toward the vegetal pole. So it seems like all over here there is a signal of fibronectin guiding these cells to go to the opposite end. So it's always easier to go through a recent paved highway, right? You go like instead of a highway that is made of pebbles, okay, or gravel. So it's always easier. Yes.
Is fibronectin marked as red in this picture?
It's not, we're gonna see it in the next image, okay? But it's normally on this edge over here, okay? We're gonna see it in the next image. So let's just let me change my image here. Here is what we see it, okay? So fibronectin, this is a sagittal section of Xenopus, early embryo, okay, right here. And we see how this fibronectin lattice in the blastocele roof is identified with this label in the little yellow greenish color here, okay? So it's at the beginning, it's over here, and then you see how it goes all the way around. So this is the fibronectin that is telling the cells to migrate on that road, okay? So all the arrows is showing that one. Is that clear, Elena? Elena?
Yeah.
Okay. If it is now clear in this image.
Oh, yeah. Yeah.
Okay, perfect. So the blastocele is indicated by this BC. This is the blastocele, and we see how as cells are epiboly, it gets displaced in this section. What we see here in B is a high magnification of this non-involuted marginal zone, okay? So this is all this section here. This is the upper following row of cells are moving, okay? And this is the lamellipodia that they create a connection and destroy the connection so that the cells can lead and start moving all around, okay? So the leading cells row, this is the FR1 and FR2. All of these cells are the leading ones when start going inside, okay? Going to exert a tractional force, and fibronectin is the surface that they are capable of following. So they're going to act as a pulling. This is the upper following, and this is pulling. All this is the fibronectin pathway that they are going to follow using that lamellipodia. So they need to create and disintegrate the binding so they can progress into the embryo. So this image here allows us to see the cell migration while the mesoderm is involuting inside. So we have ectoderm in blue, mesoderm in red, and endoderm in yellow color. So this figure clearly shows that the leading row of the cells at the front of the pack, which means the bottle cells, right? We see here the path of fibronectin and the lamellipodia affinity with that fibronectin. So it's a complex, okay, that attracts these cells to start moving, and this is the section. Now, we have already talked about catenaries, right? So to be able to do this pull interaction, we need of catenaries. We have talked about it. These are cell addition molecules that allow these filament complexes to find a leading towards the other end where they need to go. So once again, cadherin, in this case, are cadherin keratin, excuse me, and some intermediate filaments that allow to pull these leading cells. So the involuting marginal zone, the one that started going inside in here, this is after the involution through the blastopore. So this is the blastopore lip and here. And here we have the involuting marginal zone cells. We see how they start rolling over the section to form that ankerteron. Now, they do this also by convergent extension. That was another movement, cell movement that we talked a couple of weeks ago. So in here also we have the anterior population of cells migrate towards the animal pole. So this is in the marginal and it goes into the animal pole. And this is what this is showing here. Okay, it's going all the way up in this end, and including the bottle cells to form the roof of the archenteron. So once again, this is the archenteron and this is the roof of that archenteron. So the movement of these cells is coordinated by several signals, as we saw. So we have the extracellular fibronectin matrix that we talked about 2 slides prior. We also have another factor that is called the planar cell polarity, the PCP, planar cell polarity, which is normally initiated by the winglets, WNT. Okay, so we also found the caderns that gonna encode for cell addition, and we're gonna have the paraxial protocaderin that allows that cell addition so we can progress the cells. Okay, so the unfertilized egg has a polarity along that animal-vegetal axis. Thus, the germ layers can be mapped onto the oocyte even before fertilization occurs. The surface of the animal hemisphere will become the cells of the ectoderm, and we know that the ectoderm is responsible for the formation of the skin and the nervous system, while the vegetal hemisphere forms cells of the endoderm, gut, and associated organs. The mesodermal cells form from the internal cytoplasm around the equator. So this general map normally is thought to impose upon the embryo and have 2 main functions. One is to differentiate into the endoderm, and the second one is to induce the cells immediate above to become the ectoderm. So the mechanisms for the specification of these parts normally resides in a set of RNMAs that are tethered to the vegetal cortex only. And this is what we have in here. So we have the transcription factor VegT, VEG because it's placed primarily on the vegetal hemisphere, okay? So it becomes really predominant in this vegetal pole during cleavage. So as the cells start dividing, VegT is really expressed into the vegetal hemisphere. And this is what is important in the induction of the endoderm and mesoderm, as we see in the image. So this is an untreated embryo, animal vegetal pole, start dividing, VegT gets expressed greatly into this section. And then we have epidermis, a nervous system, endoderm with high quantities of this, and then the mesoderm that finds themselves between the 2 layers here becomes the mesoderm and some neurons and interneurons. If we deplete VegT experimentally, this is what we have here in this image, this results into the animal pole cells producing only ventral epidermis, okay? The equatorial region produces epidermis and neuron, but it's missing the mesoderm because only this one has the competence to induce the formation of the mesoderm. So the vegetal region usually produces the endoderm, but in here we have ectoderm and a little bit of mesoderm. So we are completely lacking of that endoderm. In addition, we have another factor that is the VEG1, which we can see here. Okay, so this VEG1 mRNA has been stored in the vegetal cytoplasm and is also translated. The production of this VEG1 factor, this is another nodal-like type protein, okay, is required to activate other genes on the dorsal mesoderm. And here we see that cascade over here. Okay. So in here we have that all these movements, cells interaction and induction of transcription factor during blastulation are really important in order to form the germ layers and specify them. Okay. So we have three things that are important in here. The first one is that the vegetal pole is specified as an endoderm by the transcription factor that is called SOX17. The second thing that is important is that the equatorial cells, remember the ones for which we start the invagination, okay, are specified as a mesoderm by transcription factor that is called eomesoderm. And third is that the animal capsules become the ectoderm. So we have a huge cascade here between nodal type, okay, but all starting with the VegT that is expressed in the animal pole and hemisphere, an expression of nodal, and this one also going to express VEG1 that will specify which the formation of the endoderm and of course the mesoderm. So all of these are important. At the blastula, a little bit more into the late blastula, not gastrula yet. So that VEG1 mRNA is translated into VEG1. This is what we see right here. Okay, and that induces the future dorsal mesoderm to transcribe genes for several wingless antagonist. So VegT activates nuclear genes that encode nodal proteins, as we see in here. Now we have nodal completely expressed in this bottom part, so the vegetal, which is the endoderm. And all of these are the TGF and beta family members that activate the expression of transcription, as we have seen prior. Now, we have a progressive determination of the amphibian axis. Vertebrate axis arise progressively through a sequence of interactions between the neighboring cells. So we're going to have a little bit of a conditional determination, right? Because the neighboring cells are telling, based on their position and the concentration of these gradients, what part of that embryo they need to form. So that inductive interaction among neighboring cells established the axis, and it was demonstrated by an experiment that deserved the Nobel Prize in 1935 to Hans Spemann. And this is what we see in this image. So Spemann experiments started a little earlier of the 1930s, but it was in 1903. And he demonstrated the nuclear equivalence. So in A, we have an egg at a stage 8 cell, okay? And in here, what he did is, the story says that using the hair of his daughter, he created a ligature in the middle, right here, okay? And the nucleus were restricted only to a half of the embryo. This is just cytoplasm, and the nuclei were only in this end, okay? Cleavage on the side reached the H-tailed cell, while the other one remained undivided. So this one divided, but this one no, which means only the nuclei were able to divide. In B, what he did is at the stage of 16 cell, a single nucleus escaped to this other end, and the ligature was tightened. And at the end, we have two complete separate individuals. Okay, and this is what happened 148 days over here. Two normal embryos were divided. So even though this one here only had one cell, because it had the same information, it had all the material that was required for the formation of a full, complete individual. Spearman performed other similar experiments that show the importance of the great crescent cytoplasm in development. Remember, the great crescent forms at the opposite end of the entrance of the fertilization because it does a 30-degree variation movement, okay? So when the zygote was divided along a plane where only one cell had great crescent material, the side without the great crescent material produced a mass of smush, an unorganized tissue lacking dorsal structure. So it was what it called the belly piece only. So here we have our egg. This is our great crescent. If we divide right in the middle, each section has part of the great crescent. We have two normal developed individuals. However, if we cut it in a different way, when only one section has that great crescent, the section lacking of the great crescent was just like a belly piece, but did not form a complete. So from here, let him believe that something in the great crescent is essential for the normal development of the embryos. So the egg fate map gives a clue because the great crescent region gives rise to the cells that initiate gastrulation. So those cells that start invaginating are part of that great crescent. So that is where the dorsal lip of the blastopore initiates, okay? So the cells of the dorsal lip are committed. And remember, committed means that they're gonna follow a fate, right? To invaginate into the blastula. And that is what initiates gastrulation and the formation of the notochord. So in 1918, Spearman showed that enormous changes in potency occur during gastrulation. So the cells of the early gastrula were uncommitted. Early gastrula are uncommitted, and the cells of the late gastrula were determined. So this is how Spearman showed that there is changing tissue between two units. So we have here, oh, my little arrow move. This is the early gastrula, and this is the late gastrula. We say that in the early there is uncommitted, while in the late there is a commitment, there is definition of the tissue. So here in A, what we have is when presumptive neural, so this in red here, tissue was transplanted into the belly area. So this is here. The ectoderm formed epidermal cells, which is what we have here. Okay, so the cells of the early gastrula were not committed to a specific fate because this should be neural ectoderm, but it formed epidermis, right? So the cells were said to exhibit conditional development. Remember, conditional is a regulative or dependent on the environment in which they are placed. Okay, because their ultimate fate depends on the position in the embryo and not the origin. Now, here in B, this is a late gastrula. So we do the same thing. We take a little bit of this presumptive neuroectodermal from the late gastrula, and we place them in the belly section. And what we have a second neural plate. Okay, so these cells exhibit the autonomous development, which means that is independent of the environment in which they are being placed. So their prospective fates was determined, and the cells developed independently of the embryonic location in which they were placed at this point. So early gastrula cells are not fully committed. In late gastrula, as the gastroceles are forming or the acantherons are forming, cells become determined. That makes sense? That is clear over there? Yeah, okay. So in 1924, later on, Spearman and another person, that is Hiral Mangold, showed that all of these tissues in the early gastrula, only one has its fate autonomously determined. Anything else can vary, but only one tissue has their fate predetermined. And this self-differentiating tissue is the dorsal lip of the blastopore, okay? The tissue that normally derivates from the great crescent cytoplasm. So the dorsal lip originates from that section of the great crescent. So how they did that? So we have here in A blastula, okay? We take here, this is the dorsal blastopore lip. We take it from here, we implanted it into another one, but where the presumptive epidermis is, okay? So that is the belly section, ventral part of this other embryo, okay? Another gastrula. And we know that it's a gastrula because we see here the invagination, the initial invagination. So this is where the dorsal lip of the blastopore of this gastrula is, okay? So it not only continued to the blastopore lip, but also initiated gastrulation and embryogenesis in the surrounding tissue. So here we have, this is where it was implanted. Later on, we see that we have a secondary invagination axis. So we're gonna have a two belly-tied individuals. This is what we have in CS. So we have two conjoined embryos that form instead of one. So the donor tissue invaginated, forming a second acantheron. It continues cell differentiation into chorda mesoderm, mesodermal structure, as it normally would have done if it was left in that embryo. So with this, years later in 38, we refer the dorsal lip of cells as the organizer, okay? And we call it the organizer because they induce the host, the ventral tissues, to change their fates to form a neural tube and a dorsal mesoderm tissue that it will become later, as we're gonna see, the somites, another transient structure, very important in embryogenesis. The second reason why we call this the organizer is because they organize host and donor tissue into a secondary embryo with clear anterior-posterior dorsal ventral axis. So if we go back, it has the whole thing, anterior-posterior, dorsal, ventral. Okay, so those two reasons make that the dorsal lip cells are called the organizer. Spearman and his students' later work showed that the dorsal lip does not organize the entire embryo, however, but rather it initiates a series of sequential inductive events. So they just need the little push, right? For all of the rest of the cells to say, oh, okay, okay, you moved me out of my comfort zone, now I need to do something else. So for this, it's important to recall that induction is when an embryonic region interacts with another to influence the second region differentiation or behavior, right? So I give you the signal, And now I give you the signal, now you know what you need to do. So, because there are numerous inductions during embryonic development, the key induction where the progeny of dorsal lip cells induce the dorsal axis and the neural tube is traditionally called primary embryonic induction. So we need a key induction initially. So all the cells that are passing through that dorsal lip can change their fate. Okay. Now, the experiments that Spearman and Mangold show that the cell of the dorsal lip of the blastopore and the notochord that form that dorsal lip constitute that organizer that could instruct the formation of the embryonic axis. But how? They didn't know exactly how that happened. So we have two new people, Nieuwkoop and Nakamura. They did experiments which provide the major clues into how that dorsal blastopore lip obtained its properties as an organizer. So mesoderm arise from marginal cells. We saw it when we see the cells invaginating and leading. We saw that the ones that are involuting become the mesoderm, right? So the cells at the equator were the equator, where animal and vegetal hemisphere meet. Those cells have the specific properties of the newly formed mesoderm that induce those vegetal cells underlying them. So here we have the experiments that they did. So they remove marginal cells, which is presumptive mesoderm. So we have here the embryo, normal embryo. We have the ectoderm. These are the marginal equatorial cells that normally are the mesoderm. And here we have the endoderm, okay? So when we remove the marginal cells, which is the presumptive mesoderm from the blastula, and put it alone, neither the animal cap nor the vegetal cap produce any mesodermal tissue, okay? So the mesoderm produce depend on the dorsal ventral polarity of the vegetal fragment. So that's what we have here. Animal, which is the presumptive ectoderm, and these are the endoderm, and has some factor to induce these cells. So the dorsal-most vegetal component specifies tissues having properties of the organizer that we're going to call the notochord. Okay, so this is the ones that are more towards the dorsal part. Okay. The ventral and lateral vegetal cell specify non-organizer tissues. So this is what we have to this section over here. So this section, the dorsal-most vegetal cells of the blastula, which is capable of inducing the organizer, has been called the Nieuwkoop center. So the Nieuwkoop center is created by the cytoplasmic rotation. Remember that the turning of 30 degrees that occurs when the sperm enters the egg. So this is the section where that turn happened. And this is the section where the great crescent is formed, okay? And this is then, the vegetal most is the Nieuwkoop center. So let's go and see what happened with that Nieuwkoop center. So we have some molecular biology in this section. So the major candidate for the factor that forms the Nieuwkoop center in the dorsal-most vegetal section is a molecule that we have seen already in the sea urchin, the beta-catenin, okay? Recall that this molecule is responsible for specifying the micromeres in the sea urchin, right? Those are the ones that start invaginating to gain the inside of the embryo, remember? So in Xenopus embryos, beta-catenin begins to accumulate in the dorsal region of the egg during the cytoplasmic movement of fertilization, that 30 degrees of formation of the great crescent. Beta-catenin continues to accumulate peripherally at the dorsal side through early cleavage. During late cleavage, the cells containing beta-catenin are located in what we call the Nieuwkoop center and organizer. So I'm gonna come back. I'm gonna go to the next slide to come back 2 seconds. So this is the disheveled proteins at the vegetal part. As soon as the sperm penetrates, we have the rotation of 30 degrees, and now this is the section where the beta-catenin gonna start accumulating. Okay, so we have disheveled, that is the main receptor in here. So, coming back. So, beta-catenin is necessary for the formation of the dorsal axis, which is really important here. If we do a depletion of beta-catenin transcripts, we're going to lack of all dorsal structures in that embryo. Conversely, if we inject beta-catenin into the ventral side of the embryo, we're going to induce a second organizer and then a second axis. So how beta-catenin become localized in that vegetal section? There are 3 proteins that seems to be responsible for the localization of the beta-catenin in the embryo. One of them is WNT11. Then we have the GSK3, another protein that we have already mentioned before, is a binding protein. And then we have disheveled. Okay, so we have those 3. We have the royal weed. Researchers have made different experiments that suggest that beta-catenin is initially synthesized throughout the embryo, but is mainly degraded by the SK3, the glycogen synthase kinase that we have mentioned before. Okay. So the critical events for the axis formation may be the movement of inhibitor of this GSK3, this glycogen synthase 3, into the cytoplasm to the opposite entrance of the sperm. Okay. And this agent may be the disheveled protein. This is the WNT pathway, which is a common suppressor of the HSK3. And we saw that when we were describing the pathways. So here in A, you have disheveled, and GBP protein is associated with the kinesin. So we have here kinesin, the GBP, and the disheveled. The 3 of them are associated together. They are mainly in the vegetal pole of the unfertilized egg. WNT11, which is the one that we see in here, is also in the vesicles at the vegetal portion of the egg. Okay, so all of this is together. There is not like they are dispersed everywhere. They are all located here in the vegetal pole. So after fertilization, we see that dramatic turn 30 degrees, and the vegetal vesicle translocate dorsally. This is ventral, this is dorsal. Okay. And here is where we have the great crescent. So the cortical rotation, which is kind of a slow, form the transportation of the fast-track microtubule, right? So this is the microtubules that attach. This is the fast transport of microtubules. And then here in D, what we have is WNT, disheveled, and GVP are released from the microtubules and are distributed in the future third of the one cell embryo. And that is exactly where the dorsal lip of the blastopore will form. Are these movements clear with these factors there? I see Emma just paying attention, just looking at.
Sorry, can you please repeat that area with VNT11, DSH, and the GBP? Is that dorsal or ventral area?
It's dorsal.
Dorsal. Okay, then I got it right.
Yeah,
Thank you.
initially is on the vegetal hemisphere, but as soon as the sperm gets in and we have the 30 degrees rotation, they get in here into the microtubules and get rotated and pass through that section of the dorsal side. This is ventral, this is dorsal.
Mm-hmm. Yeah, I pretty much got the rest. I'll add the microtubules now. Thank you.
Yeah, exactly. Okay, so now we have all of this expression in this section where the dorsal lip of the blastopore will form. So in here, the dishevel and the glycogen binding protein bind together, and those two will block the GSK3. And this is what we see here. So here we have WNT, dishevel, and GBP will bind together, and they're going to block the GSK3. And that inhibition here in this dorsal section prevent the degradation of beta-catenin in that dorsal side of the embryo. Okay, so by blocking the GSK3, which normally degrades beta-catenin, WNT is maintained and stabilized, okay, in this dorsal section. And then in E, what we have is we see the same thing as we saw in the C version embryo, remember? Here we have the beta-catenin, the nucleus of the blastomers in the dorsal region of the embryo receive the beta-catenin. We don't see anywhere else in the ventral side, but is in the dorsal side of the embryo. We see that beta-catenin that is accumulating into here, and that's why we think that these three factors here are blocking the GSK3 that normally will degrade that beta-catenin. So everything gets accumulated into that dorsal section that will become the dorsal blastopore lip. So here, once again, we have that beta-catenin accumulated, and we have the VegT and VEG1 that we have already seen in the endoderm section. Now, there are another couple of things that happen in this organizer. At the blastula stage, normally nodal-related proteins are largely produced by beta-catenin. Okay, so here in B, this is a blastula stage 8. Okay, we have VEG1 and VEGT are in the vegetal hemisphere. Here in C is a stage 9, a little later. Okay, we see that beta-catenin in the most dorsal blastomere, which we call the Nieuwkoop center, right? Right here is where we say the Nieuwkoop center was located, will cooperate where they both overlap with the VegT and the VEG1, okay? And we're going to have an activation of the Xenopus nodal-related genes, which are the XNR, Xenopus nodal-related genes. And we see that the concentration is greater, or the activation is greater, right where the beta-catenin overlap occurs, okay? So that creates a gradient being higher in the dorsal region compared to the ventral portion of the embryo. In stage 10, as we see in here, the mesoderm is specified by this greater concentration of the Xenopus nodal-related genes, okay? We also have a gradient here with the BMP4, the bone morphometric paracrine factor 4, and WNT8 of Xenopus. Okay, so we have greater concentration of this more in the ventral portion that is on the dorsal. So we see the dark color versus the pale color here. So greater concentration in the ventral side. Okay. So mesodermal cells with little to no Xenopus-related nodal genes have higher levels of BMP4 and WNT. So these are the higher, and that is the ventral mesoderm. Mesoderm cells with intermediate amount of Xenopus nodal-related become lateral mesoderm. So all over here will become lateral mesoderm. And mesodermal cells with high concentration of Xenopus nodal-related genes, and normally this is we have seen it before, and is the goosecoid, okay, then becomes the organizer, so higher concentration of the Xenopus nodal-related genes, but less of the BMPs and WNT8. So beta-catenin, really important for the expression of nodal to become the organizer, and these two will overlap with beta-catenin to have the final formation of that organizer. Function of the organizer. So we know that while the Nieuwkoop center cells remain endodermal, the cells of the organizer become mesoderm, and they migrate underneath the dorsal ectoderm. So the dorsal mesoderm induces the central nervous system to form various things. So we have the properties of the organizer is the ability to become dorsal mesoderm, the ability to dorsalize surrounding cells into paraxial mesoderm. And these paraxial mesoderm are the somite formation, right? They also have the ability to dorsalize the ectoderm and become the neural tube. And finally has the ability to initiate the movements of gastrulation. So if that zone, the organizer, is taken away, will not have the formation of an embryo, because any of the induction pathways that are necessary for the different movements involved into gastrulation won't be there. Okay, so the formation of the anterior posterior axis follow the formation of the dorsal ventral axis. Once the dorsal portion of the embryo is established, that's the first step. The movement of involution of the mesoderm will establish the anterior posterior axis. So the mesoderm that migrates first through the dorsal blastopore lip will give rise to the anterior structures. The mesoderm migrating through the lateral and ventral lips form posterior structures. So it is thought that the cells of the organizer ultimately contribute to 4 different types of cells. Pharyngeal endoderm, the head mesoderm, which is the precordial plate, as we want to see later. Dorsal mesoderm, and this dorsal mesoderm is mostly chorda mesoderm, as we saw prior in one image, that will become the notochord. And the 4 types of cells are the dorsal blastopore lip. So the first cells to involute, the pharyngeal endoderm, precordial plate, induce the forebrain and midbrain. The dorsal mesoderm induces the hindbrain and the trunk, and the dorsal blastopore lip forms dorsal mesoderm and eventually will induce the tip of the tail. And if things were not complicated enough, already. In here, we see a complete mappage of the effects of BMP, the bone morphometric proteins. So the nervous system forms from a region of the ectoderm that is protected from the epidermal induction of the BMP inhibiting molecules. Okay. So the ectoderm is induced to become epidermal tissue by binding bone morphogenic proteins. So the organizer tissue act by secreting molecules that block BMP. So here we see is block, block, block. So they are blocking the BMP in this section, and that's the organizer. So they start acting by blocking all these BMPs and allowing the ectoderm protect by these BMP inhibitions to become neural tissue. Remember, it's in this section that the cells are going invaginating and from migrating. Okay, so we need to block that in that section. So there are three major inhibitors, and are the ones that we have in here. So the first one is noggin. Okay, so noggin is a secreted protein that is able to accomplish two major functions of the organizer. The first one is that induces dorsal ectoderm to form neural tissue. And then it dorsalizes mesoderm cells that will otherwise contribute to the ventral mesoderm. The second molecule is chordin. Chordin activates beta-catenin. It binds directly to BMP4 and BMP2 and prevents the complexing with the receptors. The third molecule is follistatin. Follistatin is an inhibitor of activin and BMPs and causes the ectoderm to become neural tissue. And last but not least, we have norrin. And norrin is present in the egg, mainly in the animal pole. So during gastrula, norrin normally is incorporated into the dorsal animal cells that will become the anterior dorsal part, so the head ectoderm. And it seems to block BMP in the cell autonomous manner. So all of that is just to keep this side free of the BMP, so the cells start gaining the inside, can accomplish their function. During neurulation, that is the next step of gastrula, the forebrain, the hindbrain, and the spinal caudal regions are organized along the anterior posterior axis. So the organizer not only induces the neural tube, but it specifies which region of that neural tube it needs to form. And this is when a lot of things can go wrong. So in this figure, we have the regional specificity of induction that was demonstrated in newts by doing a transplantation of different regions of the archenteron roof of late gastrula. So remember that late gastrula cells are already committed. Okay, they have to find their fate. And they pass that into the blastoceles of the early gastrula embryo. And remember that the early gastrula embryos, they have not defined their fate. Okay, so here in A, we have the most anterior portion of the archenteron roof induce balancers. That's what we have in here, okay, which is the oral apparatus. In B, here we have the next most anterior portion of the roof induce the formation of head structures. So here we see a second head forming. In C, we have the third section, and that induce the hindbrain, because that's the portion that it should be normally. And in D, the most posterior section induces a second trunk. So, and a little bit of a tail in that section. So we have that the organism not only induces the neural tooth, but it specifies which regions. Remember, these are defined already, so they only gonna be developing the structures that they have committed to develop, even if we transplant into an early gastrula individual. So there are different types of research that shows the existence of two gradients in amphibian embryos. One that is the dorsal gradient of neutralizing activity, and there is a caudal gradient of posteriorizing activity. So two, dorsalizing and caudalizing factors. The neutralizing activity comes from the organizer and induces the ectoderm to be neural. Chordin, remember chordin, the molecule that we just mentioned, and also noggin are examples of neutralizing factors, and these are secreted by the organizer. So we have different IGF. These are insulin growth factors, okay? Also has the anterior neural development. Then we have inhibition of BMP4 signaling, normally gonna cause cells to become neural. But their fates along the anterior posterior axis will depend on the relative position of this axis to W and T. So once again, in development, and that's what you're gonna see through the courses, that we require more than one signal. It's not that just because we have one that everything is okay. We need constant feedback to say you gotta become this. It's a whole signaling stuff. The posteriorizing activity originates in the posterior of the embryo, and normally the gradients are weakened anteriorly, right? So candidates for the posteriorizing factors are W and T, okay? There's a five of the paracrine, the FGF, the fibroblast growth factor, and also one that is important, we have already mentioned it before, we wanna see it again and again, retinoic acid is really important at the posterior part of the embryo. So these two signals, the dorsal and caudal gradients, signal system, they work independently from one another. But at the same time, they have a little bit of a feedback when the gradient is slow or very few. So there are two gradients illustrated in this figure. We have W and T, and we have the BMP gradient. So BMP gradient specifies that dorsal ventral axis, while the W and T gradient specifies the anterior posterior axis. So keep this really in mind, okay? BMP is dorsal ventral, W and T is anterior posterior axis. So here we have anterior section, we have here posterior section. So in here we have in A, okay, W and T signaling pathway posteriorizing the neural tube, so anterior, posterior, okay. And we see here that as time passes by, normal density, we're gonna have a variation. It's stronger here and weaker in there, okay. So gastrulation, in gastrulated embryos, excuse me, these were stained with beta-catenin, and the density of the stain compared between the regions, and that's where we see different colors in this section. So gradient of beta-catenin, the presumptive neural plate. And this is how it goes, higher and little, okay? So in this other image over here, we have that interaction between the two. So as cells start involuting, creating that archenteron, we see higher concentration, smaller concentration, retinoic acid more in the posterior portion of the embryo than anterior, and so on. So BMPs, inhibitors from the organizer tissue, block the formation of the epidermis and the ventral mesoderm, and so on. So we're gonna go, as we advance into the formation of different tissues, we're gonna go back to this gradients over here. So all of this, and here we see the intercalation between both, okay? This is showing how the Hox genes gonna determine that anterior posterior section of the embryo during the formation of the neural tube. But we have a full chapter talking just about it. So for here, I just wanna mention that. Okay. Although the embryos normally of a tadpole that we have in here look symmetrical from the outside, there are several internal organs that change. So the heart and the gut normally are not necessarily just in the midline, okay? They are balanced on the right or the left side. So in addition to the dorsal, ventral, anterior, posterior axis, we also need to define right and left. So the body must give cues to which is the right and which is the left side. So in all vertebrates, and I have already mentioned this so far, the left-right axis depends on the formation of the nodal gene, okay? And this is expressed in the lateral plate mesoderm and is mainly expressed in the left side of the embryo, okay? So nodal gene is expressed on the left side of the embryo, and this is true for all vertebrates, okay? So in Xenopus, that nodal is the XNR1, as we have mentioned before, okay? So we have here in A, normally the intestines are coiled in one direction. If we do a knockout of this Xenopus, we have completely disorganized coiling of the intestine, okay? And that can be dramatic for the transition of the food through the embryo. And I'm just gonna have a little video to show, and we will be done with this. Here we go.
If we look at a cross-section of an embryo of the frog Xenopus, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called Epiboly. As gastrulation proceeds, a cavity, called the archenteron, forms while the blastocele progressively shrinks. The archenteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
And that is how we finish creating a frog. So that resumes the amphibian formation, and we're gonna tackle Thursday the fish embryo formation. Okay, so I wish you a very productive week to all of you, and see you on Thursday.
Have a great day.
You too, Emma.